Okay. Where are we? So Amanda, do you have bullet points and you wrote something down? Yeah. Okay. So then let's start by you sharing, and then
it's on a different computer actually. So I have like 2 screens.
Okay. Do you want to share?
Yeah, I can share. It's already pulled up.
And I think moving forward, we'll try not to make this more like a presentation, and we can know how journal clubs are done. Everyone brings their copy of the paper or pull it up on your own computers, and then we can all sit around and talk about the paper. So that's how we'll try to make this more like an interactive discussion.
Can you hear me?
Yeah.
Oh,
yes. And also see your screen.
It's all good?
Yeah.
Okay. Yeah, I have notes. If I keep looking, it's because I have notes, if that's okay.
Mm-hmm.
Do you want me to start?
Yeah. Well, why don't we talk a little bit generally about the paper, right? What the authors wanted to do, why they were interested in this problem, and things like that, to just set the stage for what they're trying to do with the figures. And I think it becomes pretty obvious from the graphical abstract that Cell Press now has on papers, right? So so historically, when you think about signals that are used in the body to increase feeding versus stop feeding, the bulk of the work that has been done is on hormones, right? The hormones that are produced by the gut, that when there are nutrients that come in to the body, to the gut, then the intestinal cells, stomach cells will produce hormones like GLP, CCK, that will signal back to the hypothalamus and parts of the brainstem that will tell the body to stop eating. And then on the other end of the spectrum is when there aren't any nutrients and the body senses, this is the internal state, if the body senses that there's a need for food intake, then there'll be neurons in the hypothalamus, like the AGRP neurons, that tell the body to start eating or looking for food, because you need to increase nutrients in the body, right? And most of that has been done in the hypothalamus through the hormones, right? Ghrelin that's produced by the intestine tells the body to increase food intake, that will signal back to the brain, mostly to the hypothalamus, to make the animal search for food and eat food. Here they're saying that they identified neurons in the brainstem that does the same thing. And these are not hormonal signals, but mostly neural signals coming through the gut-brain axis, through the nodose ganglion neurons, to the brainstem, and upwards, right? Is that what all of you gathered before you started reading the paper? And what they wanted to do? Yeah. Okay. Anything else to add to that? No? All right. So what did they try to do in figure 1? So the first step is to identify neurons that are positioned in the brainstem that reacts to fasting, right? Okay, So Amanda, do you want to go over figure 1?
Yeah. Okay. So they used the FOSS-trap method in order to test F1 generation mice. And so in A, you can see that they had the FOSS-trap mouse, and then they removed food for 15 hours, so that would act like a fasting or induce like a starvation mode. And then they injected the 4-OHT in order to trap the neurons, and then they returned the food, and then a week later, what they did was they did immunostaining to look at the different neurons. And so basically they found that there was strong neuronal activation in NTS of fasted mice versus non-fasted mice. So there are fasting activated neurons in the NTS. So that was what they first established. And then the rest of the figures mostly emphasize the colocalization or overlap between the trapped FOSS-D tomato neurons and other NTS neurons. And so figure 1B shows the colocalization in this. Can you see my cursor?
Yeah.
Okay. So this was like the overlap or the colocalization, and they found that there was little overlap in CCK, GCG, and POMC, but they did find that there was a good amount of colocalization between these TH neurons and the FOSS-D tomato, and TH neurons are also a subset of catecholaminergic neurons, so they called them CA in the rest of the paper. And so there was a presence of TH, and so then they wanted to see exactly where this like colocalization occurred. So then figure 1C shows the rostral, mid, and caudal NTS, which I looked up, and it's like the front, middle, and back. And they found that there was 15% TH neurons overlapping in the rostral and 18% in the mid, but there wasn't really any in the caudal, which is just telling them where these TH neurons are mostly located. And then once they established that there was TH neurons and that these were fasting activated, they wanted to see what would happen if they cut the vagus nerve, and they wanted to see if these would still be activated. So jumping to figure 1E, they showed that during fasting without a vagotomy, about 25% of the TH neurons were activated. So then what they did was they wanted to look at the activation of CA NTS neurons and their reliance on vagal afferents. So here you can see that they vagotomized the FOSS-trap mice, and the number of fasting activated neurons was much lower, and there were no TH neurons, which you can see here in the second column, that when they fasted them and they performed the vagotomy, there was no TH activation. And then you can also see in this third condition that when they had no vagotomy but they weren't fasted, there was also no TH activation, which means that TH neurons are all fasting activated, and they need to have that gut brain connection through the vagus nerve in order to be activated.
Yeah, thank you. Very good. So I guess most of you know that when we've been talking about this for many, many weeks now, but the rostral NTS is where the taste input goes into mostly. Right? The gut-brain axis goes through the caudal NTS. Isn't it surprising that there's a lot of FOSS-trapped neurons throughout the NTS, not just the caudal? Right. So supposedly the rostral, which conveys taste information, is also sensitive to internal state information. So if you make animals hungry, then there'll be neurons that are activated by this in the rostral NTS.
You mean activated by what? Because it's a hunger state.
Yeah, by hunger state.
Do you mean we are expecting the rostral to be activated as well?
But how is that information being conveyed? From the vagus directly through the rostral NTS?
It doesn't really have to be a neuronal thing. This is the one which I think.
So, Jia, how do you think these rostral NTS neurons get their signals from?
Rostral NTS neurons.
Yeah. I mean, in the caudal NTS, I think.
What about, let's say, I don't know, it's probably a wild speculation. It just comes to my mind. Like when you are hungry, you probably so desiring some food, you know. You're like, okay, you are stimulating your brain, some pictures or even the taste. And that can come from some of higher brain areas. Maybe they're especially active during the hunger state.
Yeah.
So that it drives the animal or us to seek for the food or seek for this sensory, instead of only know that, okay, my stomach is empty. It's probably like a way to get the animal more sensitive and also more motivated towards seeking the food rewarded sensory stuff, sensory and like a stimuli. So in that sense, I don't know. That's my speculation.
I mean, most likely these rostral NTS neurons are getting signals from elsewhere, right? Not just the nodose vagal input, right? But we don't know how that's happening, and I don't think this has been really described either. When we manage to do calcium imaging in this area, we would know how the internal state affects the activity of these neurons, but this is not something that has been described.
No.
So that, to me, the two surprising things from this figure is how much activity there is based on fasting in the rostral NTS. And the other thing was that if you look at the percentage of TH neurons, especially in the caudal, it's so low, right?
Wait, percentage of
the TH neurons amongst the activated population.
Yeah.
Yeah.
And also, I'm not sure they show how much TH neurons activated.
Right.
They only show that how much of. I don't know. Oh yeah, they only show that how much of activated neurons are TH. That's a different thing.
Yes, that's what I mean. So they're, in the best case scenario, in the rostral NTS, they're seeing 15% of activated neurons being TH positive. They have 85% of neurons, or 80% of neurons, will exclude the CCK positive, but we have 80% of neurons that they don't know what they are.
Yeah.
Right. And for the rest of the paper, these 80% of neurons are ignored.
Yeah.
Right.
Well, maybe a lot of other things are happening, and
exactly. And
it's not maybe, but there's
I
more
think that's
happening.
the major shortcoming of this paper already from this figure, right? I mean, the way that I would present this it would be very different. First of all, if I wanted to just highlight the TH, I don't think I'll use red to show that what we're going to be talking about, the yellow cells, the yellow part of the pie chart, is essentially what they're going to be talking about for the rest of the paper. But our eyes immediately go to the red part of the pie chart, which is totally ignored throughout the paper. Right?
It's okay.
Yeah,
So,
but it's
I mean,
TD tomato.
Yes, but everything is TD tomato.
Yeah. But maybe they wanted to show that this is only.
I think if I were to write the paper or work on this project,
You were zooming, only show a part of it.
No, I wouldn't focus on the TH to begin with. I think I'll be more interested in the non-TH cells. But if I were to focus on the TH, I think I would have made that pie chart with TD tomato being the red being white and the 15% being red.
Or light gray or something that is not.
Anyway.
But apart from that, I think it is interesting that there's rostral NTS neurons that are activated during fasting. That's one thing, and we should keep that in mind. And the second thing is that that they focus on the TH neurons, which are a small population out of all their activated neurons.
You want me to go to the next figure? Or
Yeah, just continue.
Okay.
So next, what they did, they wanted to show the different ways of activating those CA neurons in the NTS. So they started with the chemogenetic route. So basically, we see that they injected the AV virus to the NTS, and then showing that using CNO, they are promoting feeding in those mice. Pretty basic, not a lot to say about that. Going down to C, they chose to go with the optogenetic route. Again, the same area, NTS, this time using Chen-Dobson activation. And again, they're showing that activating using light is stimulating feeding during the time of the light. And for EFG, they're just showing us what they did with the feeding behavior. So E is just the setup. F, we can see that the dotted line is the laser activation. They have, I think, 7 mice, 1, 2, 3, 4, 5, 7 mice. So each line is another mouse. And right after activation, most of the mice start eating the pellets, I'm sorry, except for one that is taking his time, number 6 from the bottom. That's about it. There's not a lot to say. It's just basically showing us that the CA neurons in the NTS are sufficient for rapidly and potently stimulating feeding. That's about it. Not too much to say about it.
Has everyone looked up the biosynthesis of epinephrines and how all these enzymes come into action?
They're similar to dopamine as well.
Starting from tyrosine to L-dopa, you make dopamine from it, right? Mm-hmm. Where DBH comes in, right?
Well, yeah, a little bit.
So why do they choose the epinephrine and norepinephrine promoter? Like, I mean, dopamine is also one of the CA neurons. Like, they can just use TH as well here. That's my question.
They could probably. And it would still only constitute 15% of the cells. That drives me crazy. Right?
Yeah, but I think that the feeding behavior, so just by the behavior and also by the amount of cells that they do see activated, it's pretty robust. So
DBH would come as upstream of NE and E, right?
Of NENE?
Yeah. Norepinephrine and epinephrine, right? So DBH should be expressed in cells that make norepinephrine and cells that make
and epinephrine.
Yeah. So this is the sum of the two.
Yes.
Is that right?
I think so.
Yeah, how you don't agree?
Of both of them.
Yeah.
I think some of us may just Google it. Anyway, so you were talking about DBH is more specific than TH. And DBH is...
No. I'm saying DBH is upstream of E and NE. You need DBH to make E, and you need DBH to make NE.
Yeah. It's the enzyme that do the biosynthesis of NE and E.
Yeah.
Norepinephrine.
Yes.
Norepinephrine.
And epinephrine.
Right.
Pathway
so
regulating.
This, in effect, is the sum of norepinephrine neurons and epinephrine neurons.
Yes.
Yeah.
I think so. Yeah.
And
okay.
Since everyone knows that what's coming up next, that they think there's a difference between norepinephrine and epinephrine. One interesting aspect of this is that when you stimulate the DBH neurons, you're mostly getting feeding behavior. Not the suppression of feeding, but induction of feeding.
Right? Wasn't norepinephrine though what suppresses feeding? So why would it have that if it's a combination of the two?
You tell me, Amanda.
I don't know.
But you do see what's happening here, right? They're showing they're stimulating in effect both cell types, and they're seeing feeding, induction of feeding.
Are there both cell types in NTS?
Yes.
Yeah.
Okay.
Yeah.
I just think that the behavior of feeding might be stronger. So it's more important to eat when you're hungry than to stop eating, I guess, to survive.
Did they say anything about the relative abundancy of each one? Like maybe there's more epinephrine neurons than norepinephrine, so it overpowers it? I don't know how that works.
Not in this figure. Not in this part of the...
But what is surprising to me here is, and I don't know if feeding is really a binary choice, right? Is it either you eat or you don't eat? In the body, probably it's not processed that way. There are many things that go into the calculation of whether the animal should go and eat or whether it should stop eating, right? It's not that there's a single cell that signals eating, and then that cell also says stop eating. But there are many, many factors that go into this. And you would think that it's much easier to induce animals to stop eating, right? If it has a stomach ache, if it has a headache, if it has dry mouth or whatever, right? There are many conditions where we don't want to eat.
When there's more danger in the area.
To inhibit an activity, a behavior, right? Let's say even when you're testing different tastants, and now they stick a probe into your brain, which causes just a massive headache. Has nothing to do with taste detection, but you now have a massive headache. You don't want to eat that. Try drinking it anymore, right? And it could happen with the feeding behavior. So promoting some kind of a behavior like this where the animal is feeding, it's much harder to do experimentally than if we say, oh, we stimulated this region and now the animals are not eating. We don't know if that's directly tied to feeding behavior. It could be any other bodily physiological function that induces it. But they're seeing induction of feeding, so that's pretty powerful. in spite of the problems with epinephrine and norepinephrine.
Yeah.
But if you look in figure F, I thought that was pretty interesting because the timeline is a minute. That bar is a minute. So you see that
Yeah, so they
sometimes it takes like 5 minutes before something happens.
it's just the one lazy mouse, I guess. I think they're also the mice are
like
not hungry.
3 minutes, right?
Yeah. Well, the first pallet that they eat is the first line.
Yeah.
So most of them eat after 1 to 2 minutes.
Yeah.
There's one that is more taking its time. And the thing is, I think those are mice that are not hungry. So they weren't deprived of food. They're not seeking it actively.
So I know that there's some discussion of this. Remind me, is this in sated animals in the light period, light phase?
Yes.
Yeah.
In the light phase, when they usually eat less.
Yeah. But
still,
I don't think the behavior is that robust, because if you look at like the drinking thirst papers, right, and they stimulate, and I bet if they were to stimulate the AGRP neurons here, that the animals will race to this device, and it'll take less than a second for them to start eating.
To start drinking, yeah.
And even eating, I think if you use AGRP Cree and do light stimulation with AGRP Cree, that would be a much stronger response than what they see here.
Maybe.
Does everyone agree? No? Alex is skeptical.
No, I'm just thinking about it, how powerful this is, since I don't know, maybe I'm still having a hard time that our N equals 7 or 11. I think this should be done on more mice.
And also the stimulation is like continuous stimulation? Or it's just like a...
I think that yes, I don't remember right now. Wait, let me check that. It's pulses.
How long does it last? 10 seconds?
5 millisecond pulses during duration, 1 second on and 1 second off.
Forever.
So, yeah, forever. So through the whole...
Forever. Yeah, through the whole time.
Okay.
So
in that case,
I thought figure F was made very poorly in what it represented, but now it gives me even more reason to complain. So that the blue shade should be throughout the period.
Yeah.
It's not just that one pulse of at the dotted line.
Yep. Yeah, it's confusing.
I thought that mice with the arrow pointing up is a poor representation of what's going on also.
No, it's not continuous. It's like time as arrow.
And I have no idea what G is. Explain G to me.
I thought about it. I was trying to figure out what's happening, and basically you have the amount of cells that are labeled in the NTS with mcherry.
Yeah.
And the latency of how long did it take the mouse to start feeding with the laser.
Yeah, so I guess these 7 mice are the 7
dots. 7 dots, exactly.
And
so the mouse
number 6 is the one that has the latency that's at the top, right?
Exactly. That's the slow mouse.
Yeah.
Uh,
And
Interesting. So why they didn't repeat it?
why show this?
or
Oh, no.
repeat,
Why they didn't repeat it? Like
repeat
they
with the same mouse
repeat it
several
not even
sessions.
7 mouse, but repeat it several times or different days, so that you have
Same mice.
latency.
Yeah, that's what
you
I
have
said.
different latency.
Mhm.
Yeah.
It's like one shot for each of the mice.
Yeah.
Did they ever encounter this device before? Are they scared?
So yes,
Does
no,
someone
so
have
they
different
were
level of anxiety?
so they were put in this chamber for 2 days, and
Okay.
while getting used to eating those specific pellets, so it's like not new to them that this is food. So I don't think they were scared or
Okay.
So
just not knowing what's happening. But
from the drawing, it seems like they're using Lex Kravitz's Fed device that measures the amount of pellets that's taken.
who is he, or what is
That's the drawing in that
the drawing of the thing?
in the cylinder.
Yeah,
There's a small box, right?
Yeah.
So are they eventually trying to confirm there's a negative correlation? It seems they're trying to say like the more cells labeled, the less time it takes for the latency.
Oh no, I think what they're trying to say is that maybe that one slow mouse
had less labeled
had
cells.
less CHR2 expression. So maybe that's why that experiment didn't work particularly well.
But then you have 2 mice with the same cell numbers, and they were fast.
Yeah, so
like the others.
I mean, if they don't have that outlier, they can probably, I can see they can draw a regression line there, trying to prove that there's a correlation, right?
Yeah.
That's how they describe the figure. And like the more cells you have, the less time it takes. So it's faster to react and go feed.
But it's
I think
not exactly
these
what
3 panels look like they fit well as a supplementary figure.
Yeah, and also why do it for that amount of time? Why not stop it after 5 minutes? Or I don't know. Why continue? If you're showing the start of the feeding, which is what's important for them.
But it did help me to look at F, because from B or D, it seems like the feeding behavior is very robust. But looking at F, it makes me think, oh, maybe it's not as powerful as AGRP neurons.
Well, it's more powerful than nothing.
It is, definitely, and it is stimulating feeding, but it's not something where you lightly activate these neurons and the animal feels a strong urge to race to the food and start eating. And also, I think a good point is we don't know how acclimated the animal is to this chamber and everything, right? So how do they know how to get food from the pellet dispenser?
They say that yes.
They were
that
trained.
They were trained for 2 days before that. Yes.
Yeah.
So what's the whole point of having EF or the experiment with E after they prove CHR2 night stimulation actually can acutely in 1 hour increase the food intake? What's the point of adding up this crappy figure EFTG?
Well, I agree, but and as I was saying, I think this F shows that this is not such a robust behavior as B or D would suggest.
So I would think, okay, it's not robust. I would think they want to show the motivation for the food. So, but then another question is, what is the better way to show the motivation for food in math?
There's clearly increased motivation to consume food. Right? The food intake, if you take them in 1 hour chunks, then there is clearly some increase. But
some doesn't really increase much though.
What doesn't?
So some of the animals just like are just above 0.2 gram in 1 hour. So there's still a little bit like the variations though. Like in the pre-stimulation states, their animal just gets high as like a 0.25, something like that. So it's kind of a little bit, I think as Alex mentioned, like some of them may not as hungry as the others. You don't know.
Oh, maybe there's, more to it. If the animal is, I don't know, as you said, afraid of something, or just ate before that, maybe stimulating feeding is not as strong as stimulating the stop of feeding.
So based on D, it seems that they stimulated for a whole hour.
Yeah, that's what you need, or we need to.
And maybe that's too much.
Why?
I don't know. No. Is there?
So F just show probably like 10 minutes. Yeah. There are definitely other.
F yes, but for D.
For D is 1 hour. So they have the capacity. They have the potential to just go on eating, right? And supposedly those like maybe 5 or 6 bouts in F doesn't give much of doesn't announce much the uptake or consumption of the pellet. Anyway, I think the point just show us like, okay, there is a drive.
We looked into this. Do you remember how big the pellets are? Isn't it like 20 milligrams or something?
The pellets are supposed to be.
It depends on the sources.
Small.
I don't know what the sources are. But that was probably like 20 milligram, which we looked at before.
Yeah.
And it definitely takes several bouts. Maybe not 1 bout to eat one.
Yeah.
45 milligram.
25 milligrams? Okay.
Yes.
All right. So for the even for the lowest one in D, let's say it's 0.25. It's 10. It's 10 pellets though, right? They're going in. We don't know if they're eating the entire pellet, but at least they're going in to get 10 pellets. So it is stimulating food search, food intake.
Yeah. At least a little bit.
Right. All right. So let's move on to the next one.
So for next.
Can you temporarily present? Okay. So give me 1 second. So now that from figure 2, it showed that activating the ENTS neurons kind of evokes feeding behavior, and now they wanted to see what would happen if they inhibit these same neurons. So in figure A, it's a diagram that shows like the experimental setup where they bilaterally injected Cre-dependent inhibitory optogenetic vector into the NTS of the mice strain with SDBH gene Cre. And for D, it's just a physical representation where the green represents the neurons that have the virus injected, and the red is the neurons that are expressing tyrosine hydroxylase, which is the catecholaminergic expressing neuron. And for C, we show that with this optogenetic inhibition after overnight fasting, it significantly suppresses feeding. and this suggests that the ENTS neurons might be necessary for these fasting-induced eating behavior. And then moving on to D, so when there is no food, the body from the gut starts releasing these endocannabinoids. And so for D, they wanted to stimulate this behavior. And it showed setup where they took the food away for 9 hours and they injected the endocannabinoid agonist Win55. And then they wanted to look at these neurons and their activity. And E is the colocalization between the same fasting activated neurons and the FOS activity. Last one is like the merging. And F is they had two different conditions. One they injected with Win55, and the other one they just had a control group where they just injected with saline, and they noticed that for these fasting activated neurons, when the Win55 agonist was injected, there was a significantly higher activity. And for H, they moved on to the behavioral section where they noticed that injecting Win55 and activating these neurons led to more consumptions, which went back to mirrors from figure 2. And then for figure I, inhibiting these neurons with the same optogenetic inhibition method reduces this consumption behavior.
So I think so far the paper is going through a very obvious path, identifying cells using FOS trap, doing gain of function, so activating those cells to see if this will stimulate food intake, and then now doing the loss of function experiment, where they're inhibiting these neurons of interest and seeing if that has the opposite effect of suppressing feeding.
Mm-hmm.
So for E, like in the trap, TD tomato parts, like they are also staining for FOS, right?
Yes, post Win, right?
So okay, so that means like they are staining for FOS twice there.
Yes, so they're marking the cells that are activated by fasting using FOS trap, and then now stimulating feeding using Win to see if the same neurons get activated. Right? Actually, John should be answering these questions. I don't know why I answered.
Yeah.
But the condition to apply the Win is different between the two. Or is the same?
Should be the same, right?
Yeah, should be the same.
Yeah.
If the idea is trying to recapitulate, to tell us that the Win injection reduced a stable expression of like a stable expression induction of C-force in a certain or kind of fixed neuron population, then yeah, you need to have
Actually, I was mistaken. They didn't trap using fasting. They trapped using Win.
Yeah, it's Win. That's why I asked the question. It's like a remove the food, inject this, and another time is just inject the Win.
But the better experiment would have been to fast and trap, and then go back with Win, right?
But is this which way? No, which injection is done during the fasting, or there's no fasting here?
I don't think there's any fasting here. Is there?
Okay.
There's no fasting,
This is,
right?
yeah,
They
probably
they're they have food, but as they're going through the trapping, they're removing food and injecting wind at the same time.
So they inject wind and the for OHT, that is the trap time, right, for the 9 hours.
Yeah.
And then they expect that some cells will express T tomato due to the injection of the wind.
Yeah.
And then they return the food so that when the mice are feeding, the FOS should be activated as well.
No, I think by the 9 hours they're expecting the tamoxifen effect to go away.
Yeah.
Right.
So then why do they inject wind at the end?
To do the FOS staining.
And
So
that,
they just want to make sure that the same neuronal population are getting activated both times.
and that was without following the removing of food, right? The food is still there, following the injection.
Yeah.
Okay.
What are they trying to say?
Right, but the better experiment would have been to do a fasting trap and then go with wind and make sure that they're both the same population.
Fasting trap.
Yeah. Like in the first.
In first graph.
Yeah.
I think probably they did.
That would have been the experiment that I thought that they did. But apparently it is.
I feel it's why not? I think that the wind is the peptides that secreted during the hunger or during the fasting, right? So they have two conditions. One is actually without food stimulation, you only have, maybe they already been fed, I think. But before this 9 hours, there's no food. Simply, there's no food. Probably like trying to reduce the. There's also some different sensitivity between the trial methods versus the FOS staining.
Yes, but what are they trying to prove here? That
I would say probably because for the sake of the following experiments in F, they find a way to show that they can capture the same or similar neuronal population that responds to the acute wind effects. That's it. And they need, for example, if they can make the trap work in 2 hours, they can make it in 2 hours. But probably 9 hours is what they find best, works best.
Yes, but they're not trying to show that trap is working, right?
No, I think so. I think that
they are trying to show that, right?
Yeah, they try to show trap works, meaning like trap can capture the population responsible for response to the acute wind in not say wind injection, but wind stimulation.
So why can't they just use a antibody towards TH and an antibody towards CFOS in one experiment instead of using the FOS trap mouse?
I think they need to show H. Are they doing the food intake there? Oh no, what? Oh yeah, I think you're right. They didn't do any manipulation here.
So when they bring in wind, they are trying to activate the neurons, right? Responsible for this.
Yeah.
Then probably their logic is they want to try to activate these neurons while inhibit them using the photoinhibition. And then you show a behavioral difference.
Yeah.
Like eventually in eye, right? They are applying the Wing, and at the same time they're applying the photoinhibition.
That's good. But what do we learn from D to F?
So from D and E, they are just trying to show there's a large percentage of merge, right? So that shows the trap works. In F, they just want to prove like the Wing works the similar way as there is kind of like a merge with the TH neurons. I don't know how to, I think that's what they want to say. Because in previous figure, they also showed like a percentage to the TH plus.
Okay. So my question is then why don't they just use a TH mouse? I think they used the TH mouse, didn't they? They used the TH, oh.
For this,
You know,
isn't it the DBH?
TH-EGFP? Anyways, it's not important. The question is, why do they even bother doing the trap here?
Because they have their minds. They probably have other ideas. They probably have other ideas to show. So D to F just show us like the Wing activate those TH neurons, which also activated by the let's say hunger or fasting.
Mm-hmm.
So that's it. But I don't think I'm not sure they really did this job.
No.
Next they tell us that the hunger induced TH activation is due to these Wing peptides. It's nothing like that. They may even just show not Wing, but like a Wing antagonist, whatever. So that you can show the necessity of the Wing pathway or signaling in the fasting induced NTS activation. They better do that.
Yeah, I agree with Xiao. I think their intention is to want this inhibition to be like specific to these feeding neurons instead of like what we discussed, like because like the animal might just feel bad and they don't want to eat. So I think that's why they try to trap this so that they want to show in a specific time, like all these neurons are activated solely by Wing. However, in eye, it's still like when they do the photoinhibition, they did them same as before. They did it on the DBH, like NTS neurons. So I don't know if they made their point.
They want to say that the Wing in wild types increase feeding, but at the same time, if you inhibit those TH neurons, DBH neurons, this effect is not there. It becomes like it's as if like Wing it doesn't work.
Okay, at this point, just a reminder, what is Wing?
Wing is
John?
like a neuropeptide.
Yeah.
It's a little bit complicated.
And the cannabinoid agonist. Yeah.
Okay, so normally where would not Wing? This is an agonist, right? An analog, right?
Yeah, it's an agonist.
It's an analog, but
so it's an endocannabinoid.
Yeah,
and
which would be secreted where?
From GI tract.
Okay.
So it's released into the bloodstream, vascular system.
Okay.
So,
and that would signal the animal to?
So I think that the absence of food, maybe the hunger, maybe the need for, seeking for food.
Yes. So what they're trying to show here is that the endocannabinoids that are produced by the GI tract signals feeding, hunger.
Yeah.
Right? And that what they're trying to say here is that that effect
can be override by this TH
that's right.
inhibition.
So that the bulk of that effect is happening in the brainstem, in the TH neurons, because when they silence the TH neurons using in panel I, the effect of Wnt in stimulating food intake goes away.
Yeah.
Right.
But their caveat is inhibits this population also could have some side effects.
Yeah.
You don't know whether it's like maybe stomach pain or other thing. But on the other hand, there's always kind of outliers in all their behavior.
Yeah.
Extremely extreme.
So what they're saying here is that from C, when there's fasting, you would expect the animal to increase food intake.
Mm.
And when you silence these DBH neurons in the NTS, they have less food intake.
Yeah.
And to provide a mechanistic view of how this is happening, they're saying that some of this might be through the endocannabinoids, because when they use Wnt, the effect of Wnt in stimulating food intake, as shown in H, disappears when you silence the DBH neurons in the NTS.
Yeah.
And
but
I don't know what D2F is, probably very useless. But that's the gist of this figure, right? Where are we? Are we on figure 3 and an hour into this? We're like halfway through the paper.
Oh my God.
Let's go to the next figure. It's good that we're discussing the figures a lot more than before, but we don't want to spend like 5 hours on this paper.
Nobody has 5 hours.
Yeah.
Yeah. If we have another pancake, probably, yes. So what the figure 4 tells us exactly is, I think answering the what Huzhen and of you think about the DBH population, whether it's a mixture of the of its downstream like says neurotransmitter tabs, and they are like a NE norepinephrine, and also E, I think that's the epinephrine or the I think it's epinephrine.
Yeah.
So they are two types, and shown in A. I think they use different things. So red, you always think is the green line, mouse line cross with reporter ANI. So they show that different levels, actually quite close, not close, actually quite caudal, I would say. It's not really like a rostral part of NTS. And then with the TH standing there and just show, I think their point is just to show that, okay, let's create only label small populations, which corresponding to the,
in figure 1, they said that there are very few TH positive neurons in the caudal.
Very few? No, not really.
In figure 1, right, Amanda?
Figure 1.
Yeah, I actually had the same question when I saw this figure because it didn't really make sense to me.
Because they said that in
there was basically
figure 1,
none.
they say that most of the TH positive FOS tract neurons, 15% are in the rostral. What percentage was in the caudal?
18.
Like 5?
Oh, 18 in the middle, and then like none in the.
Yeah.
Oh, 9 in the caudal.
Yeah. So then here they say that they're looking at
maybe among the very few.
Yeah.
There's a lot of any. I don't know. I had the same question.
Okay.
Yeah. So they start off with a lot of promise in figure 1, and they decide to concentrate on the TH, which I don't know why, because I already thought that was strange. But now they're making all these mice, the net mice that they made, the net cree, net flip. They made at least 2 mouse strains. And I found the results to be very disappointing already. By this point, I was thinking they're digging down the wrong hole. Right? Because this well has no water in it.
You mean to further focusing on the lead population or NE population or E population is the wrong path?
Well, because they said that the TH population in the caudal is very low. At least from the neurons that they thought were activated, right?
Yeah, during for the activation, probably yes.
Yeah. So then, but isn't that what their premise was? That they're looking at the cells that are activated during fasting because they want to identify the feeding neurons?
Yeah.
Yeah, but in the caudal they saw very few. And here they're saying that now they made these fabulous mouse, which in the end turns out not to be very useful. Right? All right, but
so
go on, Jeff.
Says you are said you are have a suspect on. They said the lead cree actually labels the majority of middle NTS and rostral NTS. This is what you suspect.
No, I'm not saying that. I'm saying that the NE population, which they started off with in the beginning, is not the same population as what they're talking about right now.
Yeah, right now for the behavior.
Yeah. Because they said in figure 1 they said that the NE population, the TH population in the caudal is very low. Right? And now they're saying they're using this net cree line to activate the NE population in the caudal
part,
which doesn't correspond to the false tract neurons.
I don't know why they would do that, but the graph is a percentage, so maybe from the very small percent, it's just a proportion thing. Like it's not.
So in
there's
figure,
like a lot of
figure 1, in figure 1E, they say that the activated amongst TH population was about 30%.
Yeah,
right?
Like 25, yeah.
Yeah, or 25.
Yeah.
So that means that it's not that there aren't TH neurons in the caudal NTS. It's just that in their fast tract, the TH population was very low.
7.5.
Okay. So there may be a lot of TH neurons in the caudal NTS. It's just that with their fast tract, they didn't manage to label them.
In this, I think in the third graph, their tribe actually works great.
In the third,
because that level is quite caudal actually.
Figure 1?
No, figure 3. When they use the wing to stimulate those cells, the tribe actually works great. But also in that figure, the TH is not much in the caudal. There are some activations.
Yeah.
So they also vary their ways to activate those TH neuron right. So it's you have the pieces of everything together, which you can make a story. So
okay, but let's move along. I think we know the problem with this paper. They started off trying to trap the caudal NTS neurons that cause feeding, but it seems like they're not really going there.
No.
Yeah, and here you see that they made this mouse strain. They have to use it because they made this mouse strain for this project, but it's not really useful. I mean, it's useful for figures 4, 5, and 6, but in the grand scheme of the whole project, it's like taking us somewhere else.
Yeah. Okay, so B is the quantification, shows that most of those NE neurons concentrated in the caudal part,
Yeah.
which is of course different than the fasting-induced C-force
Yeah,
expression, right?
Yeah.
And then C is a way, so with this line, inject with the dread. I think this is dread Q, so accessory dread.
Yeah.
And then with this, the red line shows the effects of the dreads activation will induce the reduce the food intake.
Okay.
And
So
around
at this
8
point,
hours.
right? So far what we've seen is when you activate the DBH neurons, stimulates food intake, right?
Yeah.
So they probably said, okay, DBH, too broad. We want to look at norepinephrine.
Yeah.
So that we can show that with this net Cree animal that we spent $10,000 to make, we're going to show when we stimulate the net Cree neurons, we're going to stimulate food intake, right? And then they do this experiment with the GQ, and lo and behold, you see the opposite effect. This is when the student goes, "Oh no,
Yeah.
I'm never going to graduate. I'm seeing the opposite effect."
Yeah. That's probably they continue to do like another way to show that this is a consistent function.
Yeah. So now they go in this rabbit hole of, oh, maybe it's not the NE, it must be the E. And in fact, it's not even the E, it's mostly MPY, which has nothing to do with any of this, right?
Yeah.
Yeah. So let's just briefly go through the rest of the figures.
I think C is the light phase. D is the when D is when the animal actively seeking for food. So that means they have a stronger drive to eat the child. Yeah. And then in that case, this activation of let neurons still very robust.
Yeah.
And then what the E is they want to see. I think this is another way to show they can't simply use the let Cree, but here they have another mouse line like let flipo. So this is another recombinase similar as the Cree itself, but it has a different say cutting size. So in this way they have genetic. So they in this way they can enable the neuron on both are DBH positive and also let positive. And in this,
Amanda, do you know what's going on in this figure? In E, the experiment that they did in E. So you know how the
right
DBH
here in E?
Yeah, E. Yeah.
Yeah, the whole cherry thing. I just don't think I know enough about all of that to
So I think this is here. So M cherry is controlled by Cree. Capsus 3 controlled by FLIPO, so FLPO. And capsus 3 is a enzyme like suicide enzyme protein. So it will induce the neuronal death after several days or around several days. So in F, it's basically they are killing those lead neurons because in the C they show that lead neurons activity reduce the food intake, right? So they're saying, okay, maybe the rest is the contributing to food intake. So that's the case.
So net neurons are a subset of DBH neurons.
Yeah.
Right? So what they're doing is they're stimulating the DBH neurons, but at the same time killing the net population.
That's smart.
Right? And net makes norepinephrine. So they're killing the norepinephrine neurons, but leaving the rest out. And they're saying that that's mostly epinephrine neurons. So they don't have a way of directly activating just the epinephrine neurons.
Yeah, that's what the article was saying. They didn't have a way to like directly.
So they're stimulating all the DBH neurons, but killing the ones that are net positive.
And hoping the leftover ones are epinephrine.
Yes, and by doing this convoluted, twisted experiment, they see that when they do this, they see food intake increase.
Yeah.
Yeah.
Which is consistent with the TH. Oh, no, not TH. Is it a DBH?
DBH experiment.
Yeah.
Mm-hmm.
Okay.
So, okay.
Okay. And then they completely make a left turn and go to the MPY.
Why?
And MPY, people have known for many years that this is a food feeding stimulation signal, and that there are NPY neurons in the brainstem. This has been known for a long time also. So now they're just looking at MPY.
Okay.
Right?
Yeah. So they have the MPY, GFP, mouse line, C. Their point is just like, okay, those TH cells probably also colonize, some of them also colonize with the MPY, also co-express MPY. So they want to use this way to show us solely manipulated MPY neurons should also have some feeding effects. And they did the same similar dread activation, and does increase the food intake.
So you, when you do your NTS sequencing, it'll be interesting to see if the NPY population in the rostral NTS comes up outside of the taste neurons, right? Also, in your P2X experiment, do you see any nodose labeling? In your rabies? Do you see nodose and anything in the nodose? You must be muted.
You're muted.
In one of the mouse, I see the nodose.
Okay, so maybe there's some nodose to rostral NTS. You see the cherry in nodose?
No, the cherry staining didn't work that well, but the nodose definitely in one of the mouse, it has like a...
Has green or red?
Green, it has green.
Oh, so there's some rabies in nodose. Okay, well, we can move on. We're only halfway through.
All right, so in figure 5, they're trying, so in figure 4, they just says that NPY neurons, they are able to promote the feeding, and in figure 5, they are trying to double check. So basically what they're trying to do is a very similar idea as they did in the NET. So they try to remove the NPY epinephrine neurons from brain to try to prove that it's important, as we can see that in C and D, as they successfully removed NPY-E cells, there's no difference in the food intake anymore. And another thing is, after proving that NPY-E's neurons are important, then they ask, is NPY that is expressed in NPY-E neurons, is the expression of NPY important too? So they did another set of injection, as illustrated in E, that they are just removing the NPY expression. So they are not killing these cells anymore, but
two things I want to point out. One is, so far they don't have any proof that, so NPY seems to be a subset of DBH neurons, right? But they don't really show that NPY has anything to do with E, right? The only reason that they think that this is E is because when they kill with NET, they still see this behavior. and they're assuming that these are the NPY neurons.
Yeah.
The second thing is to make the distinction between labeling or activating neurons, like the NPY Cree, as opposed to NPY LOX, especially for Amanda, where they're removing that gene here rather than doing anything to the cells. So the cells are intact, but they're removing NPY gene so that they can't make the neurotransmitter neuropeptide Y anymore. Right?
Why is that? What's the difference there between?
So NPY is a neurotransmitter that typically signals feeding. So they want to make sure that they're activating these neurons, but what happens if that neurotransmitter is gone? Right? And the effect of that is what you see in G, the red
blue
versus the blue, right? So there's some component of the NPY signaling that is required for feeding. But even when NPY is not there, they're still seeing some activity, and they're just inferring that this might be the epinephrine effect. Right? Does that make sense?
Okay. It's not NPY, the neuropeptide itself.
Right. So
is
I mean,
the-
there is some, because the red and the blue, there is some difference.
Okay.
Even though, I mean, it's always hard to compare between animals, right? Maybe in the absence of NPY, the animal is sicker, right? Because this is NPY removed in all DBH neurons outside of the brainstem, right? In the whole animal, NPY is removed. So maybe the body weight of the animal is smaller.
Yeah.
You get smaller animals and they eat less. Yeah. So we don't
Maybe
know
they're
what
depressed.
this difference means.
Yeah, that could be.
Yeah, but their face value, it does seem like there's some effect of removing NPY.
Yeah.
Yeah.
But
Right.
you don't know what really happens.
Yeah, we don't know what really happens.
Yeah.
And the authors don't tell you what really happens. They just say that there's probably some epinephrine effect here.
Yeah.
All right. Let's
So
move
now,
on. Yeah.
Go on. They finally go to the. Who has been taken on this figure?
Nobody.
Aha.
So who wants to go over
Quickly go is like
yeah.
They're trying to selectively activate this presumptive NPYE population. And what do here?
And this is important because they're trying to say that this is a neural effect, not a hormonal effect.
Hormone.
Okay.
Yeah. So they want to make sure that this is going through the nodose, the gut-brain axis, the nodose, and then to the NTS. They want to show that these NPY or the DBH is going through the nodose ganglion, and they somewhat do, right?
Okay.
They see beautiful labeling in the nodose ganglion.
Yeah.
Yeah. And then when they activate these neurons in the NPY, then they see stimulation of feeding. Nothing to do with the epinephrine still. Mm. Yes? Anything else about this figure?
Well, but then there's no epiphysis, AE or E neurons in nodose, right? Is there any?
No.
Right. No. So they're not touching.
Actually, there
that
might be
part.
some NPY neurons in the ganglion. I don't know.
Okay.
It's possible. All right. So here they establish the connectivity with the nodose ganglion. Then in figure 7, they want to specifically stimulate
They are doing a
the neurons that are connected to the nodose ganglion.
Yeah.
And they seem to show that, suggesting that this is a neuronal effect rather than the brainstem neurons taking up some hormones and having that the food feeding effect, right? So let's just talk generally about the paper. And I think I made myself clear how much I like the paper.
It feels like voodoo, like a little bit of hand waving. That's like, we see some correlation, but we cannot say anything about anything.
Mm-hmm. I think it's also quite apparent that they invested a lot of effort into making the net mice. They wanted to make the most out of it.
So all those other flipper mice are available. It's not created by them.
It's created by them.
Okay.
The net flip, right?
Okay.
So, but if we take a step back and think about the entire project, I think from figure 1, which was quite beautiful, they could have gone from there to directly to NPY, right? And that would have streamlined the paper a little more, I think.
When I was reading it, I just thought it like flowed really well, and one thing led to another, and like they made it sound like everything they found was like for sure, and then they built on it, and then they're like for sure, but then looking at the figures, I was like, oh, not so much.
Yeah,
so
right.
The discussion seemed a lot more positive than the figures showed. If that makes sense.
Yes, but I thought actually the discussion was very matter-of-factly, and there's not really much insight in what they discuss.
Not the discussion part, it was more just like when they were talking about the procedures and stuff, and it would be like one line at the end, like this showed.
I see.
And it was like very matter-of-fact.
I thought the discussion section was very poorly written.
It was short.
Yes, and each of the subsections are just such obvious things. And I guess they didn't want to make their discussion about the limitations of the study, which are there are many. Right? All right. Well, we rushed through the second half, but I think we got what we want, what needed to get out of this paper. Yeah. And we can see that we can put fast trap to great use. Jahao.
Well,
but unless
it depends on what type of what kind of way you can stimulate those C4s in so many different ways.
Right. Yeah.
And what I feel this paper is kind of resonant with my previous like hypothesis. I didn't saw the paper before. Like there should be a way of NTS to stimulate food intake, while all the previous studies actually showed only inhibition.
Mm-hmm.
So that's actually their point. But then I think technically or experimentally, they fall short of what they want to show.
Mm-hmm.
You know, if they can actually show that, okay, there are two opposite functional pathway from vagus, like nodose neuron inputs, and they're coming from two different nodose populations. and then target 2 different populations in NTS, and that gives to either enhance of food intake or inhibition of food intake. That will be a cell paper.
Yeah.
And you can make it really, really solid.
Yeah. Yes. I think the implications that there are feeding stimulation neurons in the brainstem is interesting.
Yeah. Exactly.
But I don't know if they've really found anything.
But it seems that they are concentrated mostly in the taste gustatory part, right?
That's why UH, when UH is done with the single cell RNA-seq, we should look at the NPY neurons there.
Not sure they will be shown up with the rabies tracing though.
No, but UH plan is to sequence.
Oh yeah.
10,000, 100,000 cells, and then see which of those are rabies positive, right?
Okay.
Right. Anything else? Everyone's ready for lunch? All right.
It's a slice.
Thanks, everyone. We'll meet on Tuesday at 8 o'clock, but we'll try to come up with a better time. Our postdoc candidate or new postdoc who's joining us in October will be joining us on Tuesday.
Is there anyone present?
Yes.
Okay. Cool.
All right. Thanks. Have a nice weekend.
You too.
Have a nice weekend. Yes.
See you in the lab.
Bye.
Bye.
You're staying on, right? Yeah. So, where's the writing at? It's going well? Wait, let me stop this first.